Hello friends, a very good morning to all of you. In this video lecture, I'm going to talk about palynology. This video I'm making in a very good sunny morning. As you can see, sunlight is coming on my face. This video will be divided into several parts because it contains so many things. So for a better understanding, it should be split into parts. So first of all, definition, what is palynology? Palynology is a subject that deals with the study of pollen grains and spores particularly. But in an elaborate sense, scientists also include microorganic assemblages that come under the microscope while studying pollen samples. This might include the microfossils like the dinoflagellates or the chitinozoa, the scolecodonts, and the acritarchs. These are the acritarchs, as for example, are the chitinous part of the wall of the annelids. And they come under the microscope. The small fragments of that, those are also included in palynology. But in strict sense, it should be a study of pollen grains and spores specifically. The term palynology was coined by Hyde and William in 1944, and it was derived from the Greek word palyuno, it's a verb, which means to sprinkle. And the Greek verb, Greek noun itself, pel, means dust. So sprinkle dusts. So why dusts? Because these are the terms which came quite long back, maybe during say 17th, 18th century. So at that time, what are the pollen grains? It was not exactly known. They are dust-like substances. And Camerarius in 17th century, he in fact coined this term seed dust to the pollen grains. He referred the pollen grains as seed dusts because he found that these dust-like things, the pollen grains, they are responsible for the formation of seeds. So probably from there these terms have come. And later on, Carolus Linnaeus in 1750, he coined the term pollen afterward. So the historical background of this particular subject goes like this. A couple of them I mentioned already. Malpighi and Grew, they are considered as the founders of pollen morphology. Malpighi in 1671, in his book Anatomia Plantarum, he first described the pollen grains and their germination. And then Grew also in his book The Anatomy of Plants, he saw that the pollen characters are constant for a particular species, so they can be a taxonomic character. And then, as I mentioned, it was Carolus Linnaeus in 1750 who coined the term pollen. After that, many things were coming over about the pollination mechanism. And then the double fertilization also was discovered at the same time by Gladys in 1749. And Goppert and Ehrenberg were the people who studied the microfossil spores and pollen grains. So this is the way the subject developed. So we are talking about spores and pollen grains all the time. So what is the difference between them? Pollen grains are also spores. They are the microspores of phanerogams, flowering plants. But the spores, as we are mentioning every time here, is about the cryptogamic spores. They're the spores of the non-flowering plants, like the algal spores or the fungal spores, the bryophytes or mosses, pteridophytes, up to that. But here also, if you specifically compare microspore of phanerogams, you should compare the microspore of cryptogams also. That will be a better comparison. So after heterospory, when heterospory evolved from homospory, in homosporous condition there were pre-sporing spores. All the spores were of similar sizes and I will not say shapes, similar sizes, also of shapes, of course. And they developed, they were hermaphrodite. They developed both archegonium and antheridium in the gametophyte. But after the evolution of heterospory, which led to seed development, on one hand the megaspore evolved into ovule, on the other hand the microspores evolved into pollen grains. So what was the difference? what was lacking in those pollen grains. So those microspores rather. So the microspores of cryptogams, they always bear the aperture at the proximal position, proximal pole. What is this proximal pole? We will talk that in polarity also, but here I will mention that during tetrad stage, the four spores are attached at a certain point. That attachment point is considered as a pole, that is proximal pole, and the pole away from that, exactly opposite to that, is the distal pole. And there will be an imaginary line pole that is called polar axis that cross across these poles and through the center of this pole. So the aperture is always proximal in position in cryptogamic spores, whereas in phanerogams, they are never proximal, they are mostly distal, or they may be equatorial, or they may be globally located. And another thing, that is the mode of fertilization, mode of gamete, fusion of the male and female gametes, is zooidogamous in case of cryptogams, which means the microspore and the megaspore, they develop into male and female gametophyte first. These are multicellular gametophytes, where the antheridium and archegonium develop. If they are growing from isosporous spores, they will have both antheridia and archegonia on the same gametophyte. Antherozoids, which are flagellated, just like the sperms of the animals, they swim in the film of water to reach to the egg, which is residing in the archegonial venter. So this is the mode of transfer of the male gametes to the female gamete, and then fertilization occurs there. Whereas in case of microspores or pollen grains, you get a pollen tube formation. So tubes are siphonaceous, they are siphons. So the mode of reproduction here is the male gametes are carried by the pollen tube to the female gamete, that is the egg, and here there is no swimming or such kind of thing. So this kind of reproduction is called siphonogamy, which is different from that of the cryptogamic spores. So these are the differences of spores and the pollen grains. Now talking about the dispersal unit, here means the number of cells, how many number of cells at the time of dispersal from the microsporangium, how many cells are disseminated or dispersed during the time of pollination, you can say. So they may be dissociated at single cell level, or maybe two cells together, or four cells, many cells, like that. Accordingly, the names are like monads, dyads, tetrads, and polyads are there. If the cells stick together and they do not get separated, they will be transferred together, and that gives rise to the tetrad or polyad or dyad kind of dispersal units. But mostly the spores get separated, they become unicellular, and then the dissociation or dispersal occurs, that is called monad. That is the most common way of dispersal and the unit of dispersal, you can say. Another case is pollinia, where you get many spores stick together in a mass, and they together get dispersed with the help of insects particularly. And this is the case you get in two families, one in dicot and another in monocot, the Asclepiadaceae and Orchidaceae family, where you get pollinia formation and dispersal in the form of pollinia. So this is the last slide, or no, probably another slide of the same aspect. That is tetrads. Tetrads, spore tetrads. Spores are the result of meiosis, reduction division. So here there will be a microspore mother cell, diploid cell, that will undergo reduction division meiosis to form four haploid cells, and they remain for a certain period of time, they remain together, attached together with a wall, that is callose wall, that later gets dissolved. But at that time, the spores have that is called tetrad. At that time, they have different arrangements. On the basis of that, there are different nomenclatures, like they are arranged in a tetrahedral manner, in a pyramidal manner, in three-dimensional way. There are three cells in one plane, another cell in another plane. That is called tetrahedral. This is the most common one, the first one on the row, on the upper panel. You can see the tetrahedral. Then there is tetragonal, in which the four cells are arranged in the same plane. That is tetragonal. Rhomboidal is also in the same plane. And then there is in the second row you get the two diagrams that has been drawn that are decussate, where you get a plus or cross-shaped arrangement. Two cells like this, another two cells like this. So this is the kind of decussate arrangement, which also is found in angiosperms. And then there are T-shaped arrangement, two cells like this, and below that two cells are there just forming an English letter T like structure. Then linear arrangement. All these arrangements are found in different plant species. Like the tetrahedral you get in Fagus, you also get in pteridophytes like Lycopodium, Selaginella, tetrahedral arrangement. Then you get the tetragonal arrangement in Polypodium ferns. You get decussate arrangement in Magnolia grandiflora. You get rhomboidal arrangement in Annona muricata, and T-shaped in Aristolochia elegans, and linear arrangement in Mimosa pudica. In Aristolochia elegans, in fact, all these types of arrangements have been found. So let me stop here. I think you would like this video, and I would rather request you to press the like thumb at least, and subscribe the channel as well, so that you can and also press the bell icon so that you can get the notification at times. Thank you very much for watching this video.